Assalamualaikum. Hello everyone. We'll meet again. I'm Dr. Anis from Physiology, and this is my second lecture from this module. My lecture is on Physiology of Male Reproductive System. These are the learning outcomes. Function of the testes, spermatogenesis and its regulation, composition of semen and its abnormalities, regulation and actions of testosterone, and a little bit on erection and ejaculation. These are the male reproductive organs. I'm sure you have had the lecture in detail in anatomy. In this lecture, I will focus on the primary sex organ in males, which is the testis. Testis, as I mentioned, is the primary sex organ in males. The main functions are to produce sperm and to secrete testosterone. The cells of the testes include Sertoli cells and Leydig cells. Sertoli cells contain follicle-stimulating hormone receptors, which stimulates spermatogenesis. Leydig cells contain luteinizing hormone receptors, which is responsible for the secretion of testosterone. Sertoli cells are supporting cells in the seminiferous tubules. It is also called sustentacular cells or nurse cells. Each Sertoli cell is elongated from the basement membrane to the lumen. FSH acts on the Sertoli cells for maturation of the spermatid. Mature spermatozoa is released from Sertoli cell to the lumen. The functions of Sertoli cells include to supply nutrient and all the necessary materials for spermatogenesis. It secretes androgen binding protein, inhibin, and Mullerian inhibiting substance. The androgen binding protein is important in maintaining the supply of androgen in tubular fluid. Inhibin inhibits FSH secretion. MIS is responsible for regulation of the Mullerian duct during fetal period. Sertoli cells form blood-testis barrier by forming tight junctions together. This is to prevent the movement of substances from the blood coming into the seminiferous lumen. Other than that, Sertoli cells also phagocytose abnormal sperm in the seminiferous tubules. Now we come to the spermatogenesis, which is a process of formation of sperm cells from the primitive spermatogenic cells, the spermatogonia. Spermatogenesis takes place in the seminiferous tubules of the testes. The spermatogenic cells have cytoplasmic attachment with the Sertoli cells, and Sertoli cells supply the necessary materials for the spermatogenesis through this cytoplasmic attachment. This is the step-by-step process of the spermatogenesis. It begins with spermatogonia, which is located in the seminiferous tubules of the testes. Spermatogonia are diploid cells with 46 chromosomes. They undergo mitotic division to produce daughter cell, which is the primary spermatocyte. The primary spermatocyte undergo the first meiotic division and produce secondary spermatocyte, which is haploid cells with 23 chromosomes. Secondary spermatocytes undergo second meiotic division to produce spermatids. Spermatids will then undergo spermiogenesis, which is the maturation of spermatids to become mature spermatozoa. Spermatogenesis complete in about 70 days. It means that it takes more than 2 months to complete. Spermatozoa that leave the testes are not fully motile and not fertile. They gain maturation and motility in the epididymis. It means that testis is the site for production of the sperm and epididymis is the site for the maturation and also storage of the sperm. The sperm motility is further improved by the hormone relaxin, which is present in the prostate gland. Now we look at the individual sperm. Sperm has a chromosome which covers the head like a cap. It is an organelle which is rich in enzymes, which is very important in the sperm penetration of the ovum during fertilization. The head contains nucleus where DNA is located. The midpiece contains mitochondria, and the tail is like a wave-like flagellum. It propels the sperm forward. The rate of synthesis of the sperm is about 1000 per second or 30 billion per year. Wow, that's a lot, right? Now, let's have a look at what are the factors that may affect spermatogenesis. The most important factor is, of course, the hormones, the gonadotropins, the follicle-stimulating hormone, and luteinizing hormone. The follicle-stimulating hormone, FSH, is important in initiating the spermatogenesis. It stimulates the growth of the Sertoli cells. It induces proliferation of spermatogonia. It stimulates the formation of androgen-binding protein, and it stimulates the secretion of inhibin from the Sertoli cells. Luteinizing hormone is responsible for the secretion of testosterone from the Leydig cells. Testosterone is important in stimulating the development of germinal epithelia. It also completes the maturation of spermatozoa during spermatogenesis, and it is important in maintaining the spermatogenesis. Estrogen is necessary for the maturation of the spermatozoa. Growth hormone is essential for the general metabolic processes, and it is necessary for the proliferation of the spermatogonia. And inhibin is important in the regulation of spermatogenesis as it exerts negative feedback on the FSH secretion. These are some of the factors that may influence spermatogenesis. Healthy diet, healthy lifestyle, vitamins may increase spermatogenesis, whereas smoking, heat environment, hypoxia, infection, excessive alcohol may reduce the spermatogenesis. This flowchart shows the hormonal control in male reproduction. Hypothalamus releases gonadotropin releasing hormone to stimulate the anterior pituitary to release gonadotropins, which are follicle stimulating hormone and luteinizing hormone. The follicle stimulating hormone, or FSH, stimulates the Sertoli cells of the seminiferous tubules to initiate the spermatogenesis, and It also secretes inhibin, which exerts negative feedback on the anterior pituitary to inhibit FSH secretion. Luteinizing hormone, or LH, stimulates the Leydig cells to secrete testosterone, which is very important in maturation of the spermatozoa during spermatogenesis, as well as the maintaining the spermatogenesis. A small portion of testosterone is converted into estrogen. Testosterone and estrogen exert negative feedback on the anterior pituitary as well as the hypothalamus. What is semen? Semen is seminal fluid which is ejaculated during the time of orgasm. The volume is around 2 to 3 ml per ejaculation, and the volume is lower with repeated ejaculation. It appears like milky white secretions, and the pH is slightly alkaline. This is important in order to increase the vaginal pH, which is acidic, for optimum sperm motility once they are in the female reproductive tract. The sperm count on average is about 100 million per mil. For example, if you ejaculate 3 mil, you have expelled about 300 million of sperm. Wow, that's a lot, right? The speed of sperm swimming is about 3 millimeters per minute through the female genital tract. Once the semen is in the female genital tract, the sperm is capable to fertilize the ovum up to 48 hours after ejaculation. This is the composition of semen: 10% sperm, 90% seminal plasma, which are the products from seminal vesicles, about 60%, products from prostate gland, about 30%, and a small amount from the corpus gland. Semen contains ascorbic acid, fibrinogen, fructose, cholesterol, clotting enzyme, fibrinolysin, glucose, and others. A little bit on sperm abnormalities. A man with sperm count of less than 15 million per mil is considered sterile or infertile. Infertility is correlated with abnormal sperm morphology and low sperm motility. These are some of the abnormal sperm morphology. They can have defect of the head, the neck, the midpiece, or the tail. Now let's have a look at the androgens in males. The testosterone, which is the principal steroid hormone in males, is synthesized from cholesterol in the Leydig cells, as well as a small amount in the adrenal cortex. Majority of the testosterone is bound to sex hormone binding globulin, about 30% to albumin. There is a small amount of testosterone which is converted to estrogen by aromatase. Another form of androgen is androstenedione, which is the steroid precursor for testosterone. It is secreted by the adrenal cortex. Dihydrotestosterone, or DHT, is derived majority from the peripheral conversion of the testosterone in the tissue, for example, the skin. Only 20% of DHT is formed in the testes. Testosterone is converted to dihydrotestosterone by 5-alpha reductase. Now let's have a look at the functions of androgens. Some of the functions here we have covered in the lecture of Physiology of Puberty. During fetal period, androgens are very important in the sex differentiation. Testosterone, for example, which is secreted by the Leydig cells, stimulate the differentiation of Wolffian ducts into epididymis, ductus deferens, and seminal vesicles. DHT stimulates the common allergen to differentiate into male external genitalia, such as the penis, scrotum, penile urethra, and prostate. Androgens is very important in the descent of testes. Androgens are important in causing the enlargement of the genitalia, the penis, scrotum, as well as the accessory sex organs, the seminal vesicles, and the prostate glands. It also stimulates the prostatic secretions. Androgens, as I mentioned, testosterone is important in completion of the spermatogenesis, as well as maintaining the process of spermatogenesis. And as I mentioned in the lecture of Physiology of Puberty, androgens is important in the production of red blood cells. Androgens are very important in producing the secondary sex characteristics in male. The body hair growth, the pattern of the hair growth, the enlargement of larynx, which causes the deepening and break voice, the muscularity, the skin which becomes thicker, and the DHT which increases the production of sebum, which contributes to the acne formation. When we talk about androgens, we will think of the anabolic effects of androgens because it increases the synthesis of tissue protein, which gives the muscularity. Androgens also stimulates the bone growth. It interacts with growth hormone to increase the rate of bone growth during puberty. It causes broadening of shoulders, narrowing of the pelvic outlet, lengthening of the pelvis, as well as the fusion of the epiphyses in the long bones. Androgens also increase the basal metabolic rate by about 5 to 10%. It also has effects on the water and electrolyte balance, whereby it causes renal retention of the sodium and water. Androgens also play roles in psychology. The exposure of androgen during embryonic period causes male pattern of sexual behavior. Testosterone initiates libido, which is the sexual drive, and it also causes the erectile functions, which is the potency. Androgens causing the feeling of interest in the opposite sex. However, it also causes the aggressive behavior in males. This schematic diagram shows the effects or the functions of testosterone. The embryonic development, secondary sex characteristics, bone growth, muscularity, production of red blood cells, the plasma cholesterol, and spermatogenesis. A little bit on erection and ejaculation. Erection is a state when the penis becomes firm, engorged, and enlarged. It is initiated by dilatation of the arterioles of the penis. Following sexual arousal, the blood vessels of the corpora cavernosa will relax and open up. This causes the blood to rush in through the cavernosus arteries and gets trapped under high pressure, creating an erection. This process is controlled by parasympathetic activation. Ejaculation is the propulsion of semen from urethra out of penis at the time of orgasm. Following erection, the sphincter at the base of the penis will relax and the semen will flow into the penis. Simultaneously, a series of strong muscle contractions will occur at the base of the penis and along the urethra, and this will expel the semen from the body. Ejaculation is controlled by sympathetic activation. That is the end of my lecture on physiology of male reproductive system. I hope you enjoyed it. If you have any questions, feel free to contact me for any queries or any clarification. All the best and thank you for watching. Assalamualaikum.